to get started, I do have to give these disclosures. Johns Hopkins makes me give these disclosures, so I have to do so. The ones that are most pertinent here, of course, are I get research and consulting support from Thermo Fisher as well as Shimadzu. So Dr. Diamandis just mentioned some of these obstacles that we do have in this, which is this idea of automation and throughput. If you think about what we're used to in a clinical research setting or different types of settings, many of the folks that I work with are not mass spectrometry experts. They're not used to some of the details that are needed to go into running such instrumentation. And also they're used to being able to set things up and then walk away and not have to worry about it. And also much higher throughput. And so if you think about it, that's really one of the big obstacles that I see. The instrument vendors have done a much better job about making robust instrumentation. The software is easier to use. We're getting more consistent results. But the one barrier that hasn't really been addressed is how do we move samples through the instrument much faster. And then the last one we touch on here is also this idea of front-to-back traceability. I think that's a challenge as well. It's something that's really important. So when we think about what really slows this down, what I try to put up here is a typical workflow for analysis in mass spec. So we will get a specimen, we draw the blood, then that blood has to be centrifuged so that we can, there are some rare cases where we might work with whole blood, but in most cases we're working with serum or plasma, so we have to do centrifugation, and then there's then a processing step where things are aliquoted, and then we remove the protein or do some other sort of processing for extraction, and then there's another centrifugation step followed by transfer to the vials and then analysis. And the result of this is that we do most everything in batches, right? And these batches, we have to wait till we get to a certain threshold. But in labs like mine, we typically aren't working with 100 and 200 and 500 specimens at a time. We might work with 7. And so it can take us a while to get to a batch of 7. So we want to be able to do things a little bit differently. But that's for one type of analysis. Now we want to do lots of different analyses. So where do we have opportunities to improve? I think one of the ways that we can do this is to limit some of the process steps, or we can increase automation and capacity of the system. You can combine those two, of course. And then I think there are some real opportunities using mass spectrometry to modify our processes, or hopefully in the future the instrumentation, so that we can have traceability from the original sample all the way to the result, like we do in the other analytical methods we have in our labs. And then also streamlining data analysis. I'll focus on the first 3 today. I'm going to share some really vignettes. They're not a lot of in-depth. These are work that's currently in my lab. But the thing that they have in common is this idea of moving specimens more rapidly through the process. But we'll focus on these first 3 and won't really talk a lot about the data analysis today. So the first opportunity I want to talk about is really, I have a friend who talks about what a great system chromatography is to get something interesting into a mass spectrometer where all the fun stuff is, and I say, no, mass spec is really a great detector for the chromatography. And of course you need them both, but the first focus I want to have today is multi-channel chromatography, so it's not really a lot about mass spectrometry in and of itself. And so this is an example we have where using multi-channel chromatography we can do a very rapid assay for the drug Nortriptyline. And so in this preliminary analysis you'll see in the top panel, here we have Nortriptyline at roughly 250 nanograms per ml, which is a little bit higher than the range you would see in people. And then here we have our internal standard, both nice peak shapes, and what I really want to point out is the speed with which this is happening. This is about a 30-second time frame. And in doing this preliminary analysis, we were able to get good precision at 100 and 375 nanograms per ml, which are controls that we run in routine analyses we have in our lab. And then we're able to get, here you'll see a nice calibration curve. And so we're able to do basically a 30-second analysis. And so how do we get there? Some really basic LC conditions. I won't read the slide to you. I know it will be made available afterward. Using just a basic mobile phase A and B, we have a 120-second ramp. So the ramp program is really about 2 minutes. Now I know you're all smart folks. you're looking at it and say, Bill, you just said you did 30-second analysis and you're already at 2 minutes and you're not even done yet. So you're losing it somewhere, and that's true. So we have a 2-minute program, then we wash for 60 seconds and re-equilibrate. And we're using, in this case, we're actually using a guard column, so we're not using a standard analytical column, but we're really using it just to clean up the sample once we've done the protein precipitation. But the way we're doing this is we have a 4-channel HPLC system. And so what we're able to do is, while the total program is 4 minutes, the wash and equilibration steps, and this is something that's well established, certainly we didn't invent this approach, but by staggering these analyses, what we're able to do is get to a functional effective runtime of about 30 seconds. And if you carry that out, now you're getting close to about 100 samples an hour if you're able to set those things up, and that will help us more approach an automated chemistry analyzer that we would have in a hospital setting. Now, some of the factors, though, is that we're not doing a direct serum or plasma injection, and I think that is certainly a challenge for this, so we do have to do some batch preparation on the front end of this. And so what we do is we use a TCAN robot, and we use it for 2 purposes. The first reason that we use a robotic platform, and that could be a Hamilton or some other platform either, there's nothing special about the TCAN in this for us. But what we're able to do is we're able to, in these large batches, prepare the plates and then transfer the plates over to the instrument. And we're most interested actually in the traceability. We want to be able to go from the initial parent tube directly to the result. And by building the plate map in the TCAN and then importing that over to the instrument, we have a complete audit trail from specimen to result, which is quite important to us. And we developed this on the research side. We'll move it over to my other life in terms of automation workflow in the hospital soon. So what we get is 2 minutes to the first result by LCMS, and then we get 30 seconds to result after. And so we will wrap up this validation, and then we will publish this soon. But again, there are many people that have used multi-channel chromatography approaches to increase throughput. Some of those people give a way better talk on that topic than I do, including Russ Gran and Nigel Clarke. But it is something that I think is an important strategy to increasing throughput. So maybe a little more interesting when we shift as we go to things like direct sampling. So in here, we are going direct from a sample collection to getting the result, and there are a few ways to do that. But the idea here is now, how does that increase the throughput? If you're able to do it in an automated way, we have all these steps. You'll recognize that figure, but now we're just going from here to here. Now, one of the things that we would lose in that, of course, is if we take off our robotic platform, we're losing some of that traceability. So we do have to think carefully about how we'll capture that link between sample and, besides the manual transcription and checking. But we do think direct sampling has an approach. And people have been doing this, again, with dried blood spots and dried plasma spots, neither of which I'll really talk about today. I'm going to talk about dried urine. Now, dried urine is something we're interested in. So one of the things that our lab does is that we support HIV prevention trials. And so this is the HPTN network, which is a lot easier to say. HPTN is a global collaborative network focused on HIV prevention. And in many of the countries now, we're up to, I think the studies in the 20 plus years that the network has been active. We're into the HPTN 80s, or in protocols maybe even approaching 90. So there have been many protocols that have been done globally as well as in the United States, but we're seeing more and more of these studies being done for substance use. And we find there are a couple of ways. One is sort of the standard. If you're doing injectable drugs, those needles can be shared and, of course, pass infectious disease in that way. But we find also now that taking things like club drugs or cocaine can also encourage people to take risky sexual behaviors, and that can impact the transmission of HIV as well. And so there are more and more studies now focused on substance use. And so in these countries with limited resources, we think about the storage and shipping. Do they have the ability to freeze the urine? Do they have, what goes into aliquoting the urine and barcoding and all of these things? And in some of the trials, they had tested using dried blood spots. In some places, it worked well. In some places, the rats got to the dried blood spots, and it didn't work so well. But we thought this might work out for doing dried urine, so we wanted to test this out. And so very early on, I flipped these slides, I apologize. The study that I'm going to talk a little bit about today is going to be HPTN 074. And so these are samples for which we have from Indonesia, Vietnam, and Ukraine. And it's really looking at integrated intervention of counseling, antiretroviral therapy, and substance use treatment to prevent the spread of HIV. But what we did, the important part here is that we were able to collect dried urine paired with liquid urine samples. So we have both the standard way of storing and shipping the urine as well as this dried urine, so we can test and see if this is really going to be an actual way we can do business. So this is the slide that I was going to talk about it in the previous one, which is, so very early on in the studies we wanted to check out and see if we could use dried urine. We had a lot less infrastructure at the time, so it was a really very basic study published in Clinicum Acta, where we took filter paper strips. This was one done by Danny Lee Meany at the time, who we just impregnated urine into a filter paper strip. We had characterized the amount of urine that it would hold. It was really a nice little study she did there. And then we looked at recovery, just recovering the urine by water, and it was really not an elegant system at all. It was really, you took the filter paper and you rolled it up and you put it in the barrel of a syringe with some water, and then you squeezed it out, and that was it. And automation was the postdoc. And so they did not appreciate that, and neither did the reviewers. They felt like it needed to be more automated. So, but we did show that this was feasible. And in the time since that, there have been many other dried urine studies, both for metabolomics and drugs of abuse. So this is certainly something that a lot of folks are interested in. But the reviewer criticism is actually really correct. You know, I have a couple of thousand urine samples in my laboratory, and having the postdoc roll up the paper and put it into the syringe is just not going to work for that type of a study. And so we really haven't done anything with it for a long time until recently. So one of the ways to do this would be the direct spotting and going no automation, but just, well, I guess when you see the picture, you'll see it's sort of automated. It is an automated process, but it's not the way we think of a sample preparing robot. The other way is to use robotic sample preparation. And so the first thing we're going to talk a little bit about is paper spray mass spectrometry, which a lot of folks I know are interested in. So this is a potential solution. When we talk about paper spray, we have to talk about what that really is. And as much as I just said in the previous example how much I love chromatography, this sort of hurts me that we're completely getting rid of the HPLC altogether. I trained as a chromatographer, that's why I care. It's not that unnatural or anything like that. I spent a lot of time with those pumps and the columns and the tubing. But anyway, so paper spray itself is a direct ionization method. And it's very clever. It's and simple. It's taking a piece of paper and cutting it to a point, and then you apply the sample. So that's very amenable to dried sample things. You would then apply a small amount of solvent to the paper to wet it, and by capillary action, it will be drawn up the paper to the tip. And then you apply high voltage to this moist paper, and you generate an electrospray at the tip. So without having a fancy HESI source or without having an HPLC to direct sample in, you just get this nice electrospray at the tip of the paper, and you can do mass spectrometry. These are from review papers from Dr. Cook's lab. Graham Cooks his lab is the inventor of these technologies, and you'll see here this is an actual picture, so you can see the spray. But the thing I wanted to point out with this slide is that it can be both qualitative and it can be done quantitatively as well. At least in these proof of concept studies. So we thought it was a good candidate, so we wanted to jump in and do it. So here's what you see is these are actual pictures from my lab. We don't have two, I'm just showing angles in case you want to see what that looks like. So it is my lab, so please excuse the mess a little bit there. But what we have is the cartridges going here. It's very simple, and the reason I gave the second angle, you can see for perspective, this is the this is the direction where the LC would go, but here's the Q exactive we have. Okay. When I look at it, this is what I see. But so we have these cartridges. We want to move them through, but when I said that we get rid of the HPLC, it doesn't mean that it's like the infomercials you see late at night where you set it and forget it. You just put the sample on and you push it in. There's still factors that need to be optimized, and so we look at this. One of the things that one of the first things that needs to be optimized is the applied voltage, and it's really important to note. And I say this because we learned it the hard way. Is that this applied voltage has to be optimized relative to the alignment the paper spray source with the orifice. So that the gap that you have between the tip of the spray and the orifice can impact the optimal voltage that's used for the spray. You have to look at solvent composition. The same solvent does not work for all types of compounds. You have to think about internal standard addition. I think this is really interesting because a lot of the work that's done is by co-mixing the internal standard with the sample prior to application on the paper, which makes sense. You get a nice co-distribution, but that's not practical for the developing country sites. So you have to think, what are other types of ways you can add internal standard? We're collecting that data now to do this type of comparison, but one way would be to have the internal standard pre-impregnated on the strip if it's stable. Another way would be to put it in the elution solvent. And just have that be a background signal because you're using it for normalization. So we'll see how that works out. But I don't know that because we won't have access to those liquid samples, whether they be blood or urine when we first get them. So that's a challenge. We have to optimize the mass transitions or whether we do a full scan on a high resolution instrument. And then we have to think about the data acquisition time. So when we look at the chronogram, it's not really a chromatogram, right? So we call it a chronogram, which is ion current over the time. This is an example data that we just collected in the past couple of weeks. This is for EDDP, which is a metabolite of methadone. And what you'll see here on the top is the total ion chromatogram. So this is when the voltage starts, we collect the ions, and then this is when the voltage stops. Okay? Retention time is not really there's nothing retained, so that's not really what it is. But this is the time of the applied voltage for the wave, and you'll see here that, again, this is about a minute collection time that we used, and then you can collect the area under the curve for the analyte, you collect the area under the curve for the internal standard, and then you can use that for quantification of your analyte. The important thing to know here, again, is that it's about a minute, and if you look at that automated feeder of the paper spray cartridges, that means you get to a throughput of 60 per hour, right? And depending on the concentration range you're trying to get to and the count, you could decrease that time to about 30 seconds per cartridge, which then gets you up to a throughput of over 100 tests per hour. And so that, again, is another way that you can, provided that you're able to optimally do paper spray. I think the important thing to note is that it's not one size fits all. There are some things that work better and some things that haven't worked as well. So, but this is data that we did when we just took this. So here's our solvent, and the applied voltage is almost 5 kilovolts, 4.5. We have a ion transfer tube of 320, which that is another parameter that you'll need to optimize. And then we're able to measure these two transitions each for EDDP and the deuterated EDDP. And what we looked at is the reproducibility of these. We have 9 cartridges here. The 10th cartridge was misaligned, and so we got no signal. So we excluded that as an outlier. And what you see is that we were able to get, at 200 nanograms per mil, we were able to get a CV of about 3%. And that's directly in urine, that's not in solvent. So it's not idealized. It's not idealized from the perspective of the matrix. It certainly wasn't collected in the jungle or on the beach or desert or anything like that. Here's a sample calibration curve. Again, you can see a nice linear curve. We were able to get ranging from, I would say, 5 nanograms per mil to 500. The 1 nanogram per mil was a little low on that. So what are the next steps that we're going to do with this? Because that's obviously not completed work. I think for purposes of this study, what we want to look at, so this is, HPTN 074 is very interesting because some of the study subjects during the course of this 5-ish year study they have self-reported data and they have some on-site drug testing and other things, and they found that some of them in the Ukraine lost access to methadone. And you'd say, well, why is that a big deal? They just changed their drug treatment, switched them to Suboxone or something. But no, it was illicit methadone. So through the local measures, they were unable to get it, and so it drove different drug-seeking behaviors. And so we want to use this sort of to talk about to give it some context for that. So for this particular paper spray method, we're going to look at methadone, EDP, morphine and heroin, and then, because we think it'll be interesting, fentanyl and norfentanyl. So we want to focus it down, but it's really around these opiates that we'll take a look at. We're going to evaluate, as I mentioned, these internal approaches to internal standard addition. We want to look at stability. We have to do that. And then, as I mentioned, we do have field collected samples. And so we will take those liquid samples, we will then spot paper in our laboratory, and then we will also use the laboratory spotted at the sites, which have been collected over the past 2 years, and we will then do that comparison and see how things work out. I want to shift gears a little bit, still direct sampling, but we add an automation component to it, and this is using an automated dried blood spot autosampler from Holland, or the DBS Autosampler. And so this is 50-50 whether the video works, because my disclaimer is I use a Mac. I'm one of those people that uses a Mac, and so I don't, no, it doesn't look as if the video is going to work. But anyway, that's okay. Oh, there it is. Sorry, it's on, it's because of the split screen. All right, I'm not going to fiddle with it. Trust me, it actually moves. So what happens is this arm actually comes and picks up the card, and it will, I do have a picture of the card's pre and post spotting. The arm, it's actually not that exciting. The arm comes, picks it up, it will drop it in this slot, at which point there is a clamp that comes over the dried matrix spot. It will then wash solvent through that into the analytical system, and then it removes the card and it puts it back in its home. That's really the end of it. Now, when we look at this, one of the things that I would have pointed out is you'll see, it's a little light up here, but you can see where the spot has been clamped. So here's a blank card, and here pre-analysis, and here's one post-analysis, and you'll see a couple of punches, one in the middle of the spot and one to the side. And one of the things in the protocol it does is it does punch the blank spot. The non-matrix spot, so just blank paper in order to set a background, and then it punches the matrix. Now, why do I point this out? Because we were not explicit in our directions when we sent these cards to the trial sites. So the trial sites, in the interest of expediency and simplification, just took a transfer pipette and took the urine and drenched this entire white area. So now we have to figure out a different way to account for the background when we actually start analyzing those specimens. It's really important to have specific instructions. All right, so this is what the system configuration looks like. It is automated. We have the Holland DBSA system that is then in with a one-channel TLX system. This is TLX being shorthand for turboflow and analytical chromatography. The reason we do this is that we are incorporating the dried matrix. So there may be precipitates in there may be other matrix components that we want to make sure that we don't get onto our analytical column. So we would go from the Holland system clamp to the turboflow, and then a standard flow into the analytical column, and then using a TSQ quantum ultra. There were some challenges in terms of communication with the instrumentation. We did do a standard operating procedure for system configuration and everything. This was worked out in a three-way effort between Thermo Fisher, Holland, and Johns Hopkins. Here again are the conditions if you are interested, using the Cyclone P, and then we used a Phenomenics Kinetics column for the analysis here. The big part that's mostly of interest is that in this type of assay, it's actually a screening assay for 45 different drugs. We typically use this for subjects that are in getting chronic opioids or getting treatment for drug addiction. But this is a true screen. It is not a confirmatory or definitive test. We only monitor one transition per compound, whereas standard, typically in a quantitative assay, we would have a quantifier and a qualifier ion. And we only use 5 internal standards for process control. We do not have an internal standard for each drug. This is a qualitative yes, no. We are not quantifying the drugs. But what we need to be able to do is differentiate, because there are some isobaric compounds that do migrate at similar times. So here's the sample chromatogram. This is a chromatogram, not a chronogram. So we're back to using HPLC. What we'll see, this is an example of norfentanyl. This is where things go really well. Okay, we did not really see, this is at the limit of detection of the assay, 20 nanograms per mil, and we did not really see any drop-off in performance between the liquid and the dried urine sample, and I would love to say that that was the case across the board, but it's not. Here is what we see with norbuprenorphine. So here's our liquid sample. So we do expect when we start analyzing the liquid versus the dried urine samples that we will probably see some differences in detection, where the limit of detection is not as low for the dried sample. And then that's discounting any stability issues. We may have some stability problems as well. So I'm anxious to see how all this turns out. If we look at, the data has been being collected right now. In fact, I was emailing with Abed in our laboratory as I was walking in to sort of guide the collection of the validation data. We do have some preliminary reproducibility data on peaks, and some of the peaks, even at the limit of detection, are quite good. We could probably use this quantitatively for lorazepam if we wanted to. We would probably not use this quantitatively for methylphenidate. It does not fare quite as well in our DBSA assay. But you'll see that we have a wide range. It doesn't translate well, 45 drugs in a big table in a presentation. But so we have limited detection, mid-level, and high control samples, and you can see the pattern that you would want to see, right? Which is the CVs both for the low end and the high end do come down as the concentration goes up. So it does work out okay. So what are we going to do with this next? We have the same types of samples. So in addition to sending out paper spray cartridges to the clinical trial sites, we did send out the DBSA cards. Those are in the lab that we have right now. And so we'll look at sources of interference. We don't have to worry about the internal standard addition. That will be introduced through the mobile phase. We're not doing as much with that. We're doing some stability testing, and then we will do the same type of thing where we spot fresh cards in the lab. We'll analyze the trial site cards, and then we'll see what that looks like. That is more automation. It's not high throughput. So this throughput is closer to 10 an hour, but you can walk away from it, so that's good. All right. So the last thing that I'll wrap up with today is multi-channel automation. And so this is a fun project we have looking at plasma renin activity and aldosterone. And so this is using a Prelude SPLC system, which is still it has turbo flow chromatography as well as an analytical LC. And we have two channels for this. So we can do either we can multiplex the analysis like we did at the beginning of the presentation, or we can use different chemistries, which is what we did in this case. So what we did, because for aldosterone, we really needed to operate in negative ion mode, and to optimize the sensitivity of the assay, we really needed to do some different chemistries. So in this case, rather than doing multi-channel and increasing throughput, we're using automation and leveraging the channels to do different chemistries. So we have channel 1 is plasma renin activity, channel 2 is aldosterone in progress. So with this, I'll show you the experimental conditions, but I wanted to show the data first because the data looks really nice. It's always nice to show data that's pretty as opposed to messy. But here we have the peak for our angiotensin analyte and the plasma renin activity as well as the internal standard. And you see we have quite a nice calibration curve here. So the way that we do this is we have a cyclone SPE and C18 column. We're using an aqueous formic acid solution as well as methanolic solution. The mobile phase 3, for those of you that do turboflow chromatography, you'll recognize this as Joe's Magic Mix, mobile phase C. We use a C13 and N13 labeled internal standard. And so this is a little bit out of my range because we are now working with peptides, and I don't often do that. So this is me dipping my toes in that water. For PRA, we're doing positive ion mode, and here's the fun part of it. So folks like Brian Keevil and Dan Holmes and some others have already published plasma renin activity assays, and they're really good. So, but we wanted to try something a little bit different, which is to do it completely hands-off. So what we were able to do is put the plasma in the plate, and then we add the incubation buffer. And then what we do is we just incubate it overnight. So in this case, we would just walk away. We put the plate in, we do an initial analysis of angiotensin, we walk away, and then we would do a second one. And to accurately capture the time in terms of that, we know we'd have some variability, but we would just use the timestamps of the injection because the reaction is stopped when it is pulled from the autosampler and injected onto the turboflow column, so that's the quenching part of the reaction. We're automating the quenching, we're not adding a quenching buffer, okay? And we were able to get some samples with known values. I'm showing you these, okay? And so then we thought, well, let's compare them to what another lab got. It's not so good so far. But it's just our first effort at it. I show this because I like to be transparent. Dr. Diamandis, you will not have to be a police officer for this. This is real data, okay? Now, what was interesting to me, though, at our first try of this is that if you take the exception of a very low activity in It's consistent, and it's all about a 5-fold difference. So the question is, and we've actually talked about this, the idea of are you getting what you buy? We didn't actually verify. We're going to send out our calibrators for external verification to make sure that we have what we think we have, or that they have been made properly. It's possible that we have made a dilution error, or we didn't receive something correctly, or it may be that we need to optimize the way we're setting up our dilution and other things. This is sort of our first stab at it. We have some more development to go, so this is a story that's not finished. But I think it's kind of fun because we'll be able, from the same plate, to take in an automated way where we do angiotensin, then on the second channel we can do an aldosterone on the same sample, and then the next morning we just hit the start again, and then we get the second PRA, and we'll be able to walk out with a PRA aldosterone analysis. Okay. Well, I appreciate your patience. Again, I would second Dr. Diamando's comments, being here so early in the morning. I know that I did not want to get out here so early, but I said I would. So, and they bribed me with coffee. But I do have some thoughts, which is I would second exactly what was said in the introduction, which is mass spec has the potential to really have significant impacts both in the laboratory medicine, clinical pathology aspect, but also I think we didn't have time to talk about here, but I think there's going to be some really great things done in the anatomic pathology side of things as well. I think it's a technology that is going to be high impact in laboratory medicine. It has the benefit of you can use it with lots of different sample types, but you really are going to have to address, and you, meaning we, but also the vendor, you guys got to do some things too. There are some challenges that need to be met before this is really going to have the impact that meets the potential. And I think that's what we're really interested in doing is helping get there with the rest of the community. So with that, I do want to acknowledge the folks in my lab that did this. Autumn has run my lab for 12 years, and she's pretty awesome. And we have Abed, Sean, and Branch, who helped generate the data. Have to acknowledge my colleagues in Johns Hopkins School of Medicine. We do get great support from Thermo Fisher, so Mari and Joe helped us with some technical aspects, and then Peter from Spark Holland was great. And then these are the funding sources of the work that I showed today.